Okay, so good afternoon, everybody. I'm so happy to be here again in this new session of the International Seminar Series on liver toxicity and steatotic liver disease. For me, it's a pleasure to announce that the speaker of today's session, which is Bernard Fromenty. He's a research director at INSERM and currently co-directs one of the teams at the New Mechanism Institute in Rennes, France. Also, he has been working for 35 years on liver damage induced by different xenobiotics, including drugs, alcohol, and environmental toxins. And his research aims to determine the mechanism by which certain xenobiotics can induce different liver lesions, with a particular interest for steatosis and steatohepatitis. More specifically, his research focused on mitochondrial dysfunction, alteration of lipid homeostasis, and oxidative stress. More recently, he has extended his research to the issue of hepatotoxicity in the context of obesity and muscle D. And also, of course, he is the co-author of more than 140 original articles and review. And today, he is going to talk us about drug-induced hepatic steatosis, the role of mitochondrial dysfunction, and other alterations of lipid homeostasis. So it's a pleasure, Bernard. When you want, you can start. Thank you.
Yeah, thank you very much, Marina, for this introduction. I will just start. Don't want to. Sorry, it worked before, but yeah. So yeah, I wanted just to make some recall about hepatic steatosis. Although I know that most of you know this lesion, but this lesion corresponds to an abnormal accumulation of lipids. It is mainly triglycerides, and this lesion is deemed pathologic when at least 5% of the hepatocyte have intracellular lipid droplets within the cytoplasm. And steatosis or fatty liver is frequent in overweight or obese people, which is now called metabolic dysfunction associated steatotic liver disease, or MASLD. But as you may know, steatosis can be also induced by other causes like ethanol abuse, some environmental contaminants and drugs. And today I will speak exclusively about drugs. And regarding drug-induced steatosis, there are unfortunately not so much studies that report the frequency of this lesion. However, we can find some figures, for example, in the article from Bure in 2004, where he mentioned that about 12% of the hepatotoxic drugs can induce steatosis. And more recently, a study from Kleiner in 2014 reported that 25% of the cases of suspected DILI were characterized by the presence of steatosis. In this talk, I would like to stress that actually there are two types of steatosis, namely macrovascular steatosis and microvesicular steatosis. Regarding drug-induced macrovascular steatosis, it is the most frequent form of drug-induced steatosis. And as you can see on the slides, the hepatocytes are characterized by the presence of a large lipid vacuole, which displaces the nucleus at the periphery of the cells. Macrovascular steatosis, whatever its cause, is considered as a benign liver lesion, at least in the short term. There are possible hepatomegaly or slight increase in ALT levels. However, in the long term, this lesion can evolve towards steatohepatitis. But today, I will not talk about steatohepatitis because I will it will be too extensive for my talk. Some example of macrovascular steatosis induced by drugs is example the case of amiodarone, paroxetine, methotrexate, sulindac, and rifampicin, and I will talk about some of these drugs later on in this talk. Beside this, there is also a particular case of steatosis, which is called microvesicular steatosis, which is a rare form of this lesion, which is characterized by the presence of many lipid vesicles within the hepatocytes, and this vesicle leaves the nucleus at the center of the cells. And on the right part of the slides, you can see a picture that we have done from a recent paper from a case of a young girl with a fatal case of acetaminophen hepatotoxicity, which was characterized by the presence of microvesicular steatosis. So, I mean, this form of steatosis is a severe and potentially fatal liver disease with the presence of hepatic cytolysis, with moderate or high increase in ALT levels, could be associated with multi-organ failure, encephalopathy, severe hypoglycemia, and in some cases, as you will see, lactic acidosis. Example of drugs inducing this form of steatosis includes amiodarone, paroxetine, nucleotide reverse transcriptase inhibitors, such as AZT and D40, as we will see further, and phyarluridine. You have noticed that some drugs can induce either macrovascular steatosis or microvesicular steatosis, and I will show you some hypotheses later explaining this possibility. So before to enter into the details regarding the mechanism leading to drug-induced steatosis, I would like to recall some of important features of mitochondrial function and dysfunction within the hepatocytes. As you know, the mitochondria is here in the liver and other organs to synthesize energy. And this energy is derived from the oxidation of many substrates, including pyruvate from glucose, and also from amino acids, and also lately from fatty acids. Regarding fatty acid, this is done via the beta oxidation, via the fatty acid oxidation. And this fatty acid oxidation is leading to the formation of acetyl coenzyme A, which has two different features. It can either form ketone bodies, which are used for ATP synthesis within extrahepatic tissue, or within the mitochondria, acetyl coenzyme can enter the Krebs cycle in order to be fully oxidized into water and other reduced equivalents. It is important to remember that either beta oxidation or the Krebs cycle are leading to the generation of reduced equivalents, which are NADH and FADH2, which are very important for the synthesis of ATP. Indeed, the NADH and FADH2 are giving their electrons and their protons to the respiratory chain in order to generate ATP. Briefly, the electrons from the NADH and FADH2 are given to some complexes of the respiratory chain, complex 1 for NADH and complex 3 for FADH2. And this electron circulates along the respiratory chain up to the complex 4 of this chain in order to combine with oxygen and protons in order to generate the water. Along this circulation of electrons, there is an ejection of protons, which creates a large membrane potential, which is used by the ATP synthase in order to transform ADP into ATP. And this whole system is referred to as the oxidative phosphorylation system, or OXPHOS. We have to remember that when the respiratory chain is severely impaired, this can lead to the inhibition of all the oxidation system in the mitochondria, in particular the beta oxidation and the Krebs cycle. Because when the respiratory chain is blocked, there is an increase in NADH and FATH2, and of course, a reduced levels of the oxidized components of these cofactors, namely NAD+ and FAD. And this is the lack of NAD+ and FAD, which impair the beta oxidation and the Krebs cycle pathway. I would like also to remember that within the mitochondria, there is a small piece of DNA, which is called the mitochondrial DNA, and which is very important because it encodes 13 polypeptides of the mitochondrial respiratory chain, in particular in complex 1, 3, 4, and 5, or ATP synthase. And also we have to remember that this small piece of DNA has to be replicated constantly within the mitochondria. And this is thanks to the DNA polymerase gamma. And another important features of mitochondrial DNA is the presence of several copies per mitochondria. And which means that within one hepatocyte, there is several hundreds or several thousands copies of mitochondrial DNA genome. And when mitochondrial DNA is severely altered, this can lead to an impairment of the respiratory chain, and thus an inhibition of the oxidative pathway of the mitochondria, including the beta oxidation. So I will first begin this talk regarding mechanism for drug-induced microvesicular steatosis. And I just want to make some history to remember that in the late 80s, Dominique Pesser hypothesized that drug-induced microvesicular steatosis could be due to mitochondrial dysfunction. And he made this hypothesis based on two important observations. First of all, it was known that microvesicular steatosis could be observed in different inborn errors of mitochondrial fatty acid oxidation, or also in some mitochondrial cytopathies that are the name of genetic disorders of the OXPHOS system. And another observation was that microvesicular steatosis could be also observed in the Reye's syndrome, which is triggered by the acute viral illness treated with aspirin, and whose pathophysiology was known at this time to involve mitochondrial dysfunction. So, I mean, it was at this time that I joined Dominique Pesser lab, and we did some investigation regarding different drugs, and in particular regarding amiodarone, which is an anti-anginal and anti-arrhythmic drugs that was reported to induce microvesicular steatosis in a few patients, but also more frequently macrovascular steatosis. And in some reports, there was also steatohepatitis and cirrhosis as well. The structure of amiodarone is indicated in below in this slide, and you can see that it is an amphiphilic molecule with a lipophilic moiety on the left, and a side chain, which is a diethyl amino ethoxy side chain, which is rather hydrophilic. But I will come back on this later. So, I mean, we performed some experiments in mice and first measure liver triglycerides. And as you can see, the, the injection of amiodarone, a single injection of amiodarone in mice, induce after 24 hours an high increase of triglyceride, especially after the highest dose, which was 600 milligram a kilogram. And we perform liver histology after after 24 hours of this dose, and we found that there was microvesicular steatosis in the liver. In order to assess mitochondrial function, we use UC14 palmitic acid, which is fatty acid which is labeled on each carbon, and thus it is possible to assess mitochondrial oxidation of this fatty acid either in vitro on isolated mouse liver mitochondria or in vivo in mice. In vitro, with mouse liver mitochondria, the principle of this assay is to incubate mitochondria with UC14 palmitic acid for several minutes and stop the reaction with acids, and you just measure the 40 acid 40 C14, sorry, acid soluble beta oxidation product that are corresponding to ketone bodies and to some extent to intermediate of the Krebs cycle. And as you can see, amiodarone induce a dose-dependent decrease of mitochondrial beta oxidation, which was maximal after 250 micromolar of amiodarone. In vivo, we were also able to show that amiodarone induce a sharp decrease in oxidation of fatty acids, and particular palmitic acids, by measuring the exhalation of C14CO2, because mice were kept in sealed tubes, and we just recover the exhaled air and measure this C14 CO2 after 3 hours. And at this time, we were also interested by the measurement, the assessment of the activity of the respiratory chain. And at this time, the Sears apparatus was of course not available. So, I mean, we use a Gilson oxygraph equipped with an electrode, a Clark platinum electrode, that is able to measure the oxygen in the medium. And so in this experiment showed on the slides, we measured the oxidation of malate or malate plus glutamate, or the oxidation of succinate. And as you see on the graph, when you add ADP in the medium, in the presence of the substrate, you have first a state 3, which correspond to the transformation of ADP into ATP. And when ADP is completely consumed, you have the so-called state 3, which is a sort of resting state of respiration. And in this state, we apply different concentration of amiodarone. And whether it was for malate glutamate or for succinate, we observe for low concentration of amiodarone a stimulation of the respiration. For example, for glutamate malate on the left, for 20 micromolar or 100 micromolar, there was a stimulation, an increased consumption of the oxygen consumption. And for highest concentration, for 200 micromolar and 400 micromolar, there was first a stimulation, and then there was a decrease in the respiration. For succinate on the right, this was exactly the same story with an acceleration of the respiration with 20 and 100 micromolar. And for the highest concentration, 400 micromolar, there was an acceleration, and then there was an inhibition of the respiration. So, I mean, in these experiments and others that I don't have time to show, we demonstrate that low and moderate concentration of amiodarone was able to uncouple oxidative phosphorylation, while higher concentration inhibit the complex 1 and 2 of the respiratory chain. And we did other experiments with other substrates and showed that there was no inhibition of complex 3 and 4. So, I mean, with Dominic, we propose the following model in order to explain the OX-phos uncoupling. And this model was as follows. I mean, we think that amiodarone can cross easily the outer membrane of the respiratory chain. And within the intermembrane space, these drugs can be protonated on the tertiary amine of the diethyl amino hydroxy side chain. And once this drug is protonated, it is pushed by the membrane potential, and thus it can enter into the matrix. But this has the consequence of decreasing the membrane potential, and the mitochondria, in order to restore this membrane potential, has to increase the transfer of electron within the coupling site, which explain the increased rate of oxidation. Another consequence of the uncoupling of the oxidative phosphorylation is that no more proton can enter into the ATP synthase, and so you don't have any more this synthesis of ATP. Another interesting observation that we made is when we compare the curve of the inhibition of amiodarone regarding the beta oxidation of UC14 palmitic acid, and also the curve resulting of the effect of amiodarone on either malate and glutamate oxidation or succinate oxidation. Just for malate, glutamate, and succinate oxidation, please note that the graphs that are presented on this slide are in the presence of DNP, which is an uncoupler, and this actually allowed us to determine the effect of amiodarone only on the transfer of electron and not on the complex 5. So, I mean, in this experiment, we showed that we have strong and significant inhibition of beta oxidation for the 100 micromolar concentration of amiodarone, whereas we had no inhibition of the oxidation of malate and glutamate or that of succinates. And so this strongly suggested that amiodarone was able to inhibit specifically the beta oxidation at the dose of 100 micromolar, whereas for higher dose, there was probably an inhibition of beta oxidation by two mechanisms, that is inhibition of the beta oxidation itself and also inhibition through the alteration of the respiratory chain. So, I mean, we propose also with Dominique the following model. When amiodarone enter within the mitochondria, it can accumulate within the matrix, and we did some experiments in keeping with this hypothesis. And once the amiodarone accumulate in the matrix, it can first inhibit the mitochondrial fatty acid oxidation. And when the concentration are higher, it also inhibits the respiratory chain, which can further inhibit the mitochondrial fatty acid oxidation because, as I explained before, the inhibition of the respiratory chain can secondarily inhibit the oxidation of fatty acids. So, I mean, these results were further reproduced by the group of Stefan Krenbühl in Switzerland. And indeed, he published several papers confirming that amiodarone was able to uncouple OXPHOS at low and moderate concentration, whereas for higher concentration, amiodarone is able to inhibit the respiratory chain complex 1 and 2, but not that of the complex 3 and 4. And very interestingly, the group of John Horowitz in Australia published a paper in 1996 showing that amiodarone can directly inhibit CPT1, which is the carnitine palmitoyl transferase 1 enzyme, a very important enzyme of the oxidation of the long chain fatty acids. So, I mean, all these papers largely confirm our previous results on the effect of amiodarone on mitochondrial function. After the work of amiodarone, we also showed that several drugs able to induce steatosis were also able to have a dual effect on the mitochondria with uncoupling effect and also an inhibition. And we showed that with, for example, with perexilin, which is an anti-anginal drugs like amiodarone, but unlike amiodarone, this drug was withdrawn from the market in several countries because of its toxicity. We also report that for diethyl aminohydroxyhexestrol and also for tamoxifen. And as you can see, all these drugs have either binary amines or tertiary amine. For example, for tamoxifen, tertiary amine like amiodarone, that is responsible for the uncoupling effect of this drug because this binary or tertiary amine can be protonated and can be the cause of the uncoupling effect. As I introduced before, the mitochondrial DNA is very important for the mitochondrial function, in particular for the respiratory chain. And there are some drugs that are able to induce microvesicular steatosis and other severe side effects through an alteration of mitochondrial DNA replication. And this is the case with the NRTI, so the nucleoside reverse transcriptase inhibitors, AZT, zidovudine, D40, stavudine, and DDI, didanosine. And these drugs are able to inhibit the DNA polymerase gamma, which is present within the mitochondria, and below this lead to a progressive decrease of the mitochondrial DNA levels in the liver, and sometimes in other tissue like muscles. And it is deemed that when the mitochondrial DNA copies are below 40% of basal level in the liver, this will lead to a severe impairment of the mitochondrial respiratory chain, with the different consequences for fatty acid oxidation, with the occurrence of steatosis. Also with an inhibition of the Krebs cycle, which explain the lactic acidosis that can occur in this patient. And also this impairment of the respiratory chain can lead to a severe depletion of ATP in the hepatocyte, leading to hepatic cytolysis. Not only drugs can induce mitochondrial DNA replication, but some drugs can also induce steatosis by impairing the mitochondrial DNA translation. And this is the case with tetracycline and
Bernard, we can't hear you.
Can you hear us, Bernard? Could you please try to fix the sound and then go back about 2 minutes? It's still not working. Can the technical team help us, please?
Maybe you have to leave and get into again, no?
Okay. Bernard, could you log off and try reconnecting, please?
Bernard, please leave the log off and reconnect again.
Are you hearing me?
Yes, now
Yes.
perfect.
Okay. So can you tell me when
Could you
the
start on this slide again, please, Bernard? Just this slide that's up about tetracycline and linazolid.
linazolid? Okay. And so, I mean, it was okay for that?
Yes.
Yes, it
Okay,
was.
okay,
Yeah.
fine. Sorry for this interruption. I don't know exactly what's happened. Okay, so yeah, with regarding the mitochondrial DNA, there are some drugs that are able to induce steatosis and also other side effects because they can impair mitochondrial DNA translation. And this is the case of tetracycline and linazolid that have been shown to interfere with the mitochondrial ribosome. And interestingly, these mitochondrial ribosomes have a structure very close to bacterial ribosomes. So, I mean, with Dunia Leguiou, we confirm in HepaRG cells that low concentration of linazolids were able to reduce the expression of ND1. It's the panel of the right, ND1 and COX2 that are encoded by the mitochondrial DNA, whereas the expression of COX4, which is encoded by nuclear DNA, was not altered. And as you can see, the mitochondrial DNA level was not impaired, was normal with after the treatment of linazolids, and there was also either no change in mitochondrial RNA encoding for proteins, or a slight increase in MT ND1 and MT COX2. And in this paper, actually, we uncovered that ritonavir, which is an HIV protease inhibitor inducing steatosis in some patients, was able to also impair the mitochondrial DNA translation. And as you can see on the right, ritonavir, especially for the highest dose tested in this study, that is 45 micromolar, strongly decrease the expression of ND1 and that of COX2, again, encoded by the mitochondrial DNA, whereas there was no change in the expression of COX4 encoded by nuclear DNA. And again, this was not due to any change of mitochondrial DNA levels, and there was no decrease in the expression of the corresponding mRNA in the mitochondria. And interestingly, this effect on mitochondrial expression of ND1 and COX2 was associated with decreased level and activity of complex 1, and also with a decrease in the maximal respiration measure with the CROS apparatus. So when we look at all the mechanisms that can lead to drug-induced mitochondrial dysfunction and microvesicular steatosis, we can see that some drugs can can directly impair some enzyme of the mitochondrial fatty acid oxidation pathway, either by altering the enzyme itself, and for some enzyme at the right of the slides, some drugs are able to deplete coenzyme A and or L-carnitine, such as valproic acid or ibuprofen. But I didn't have time to enter into details today for this mechanism. And also, as I told you before, mitochondrial fatty acid oxidation can be inhibited because of a strong impairment of the mitochondrial respiratory chain. and also secondary to an alteration of mitochondrial DNA replication or translation. Lastly, there are some investigation showing that triglyceride is able to induce an oxidative mitochondrial damage and degradation. So just also to answer a frequent question that can be asked is whether microvesicular steatosis is a severe liver disease. And in fact, beside the decrease within the liver of ATP, which is responsible for liver failure, as previously explained, the inhibition of beta oxidation is leading to a decrease in acetyl coenzyme A, which is not only responsible for the reduced production of ketone bodies, but also for a decrease in gluconeogenesis, and thus a decrease in the production of hepatic glucose. This is because acetyl coenzyme A is an essential cofactor for pyruvate carboxylase. And as you can imagine, if you have a sharp decrease in ketone bodies and in glucose production, this could produce an energy deficiency in extrahepatic tissues, especially in case of fasting periods. So to conclude with this part, microvesicular steatosis can be considered as a mitochondrial disease. Indeed, almost all drugs inducing microvesicular steatosis in patients have been shown to inhibit the mitochondrial oxidation of fatty acids in experimental investigation, either in vitro or in vivo. And also, for some drugs inducing microvesicular steatosis, for example, this is the case for valproic acid and aspirin, some markers of mitochondrial dysfunction have been detected in liver, blood, or urine of the patient, and in particular, an accumulation of acylcarnitine and decarboxylic acid derivatives. Regarding the second part, that is the mechanism of drug-induced microvesicular steatosis, it should be mentioned that actually, because microvesicular steatosis was considered as a benign lesion, there was not so much investigation in the past because people don't care about this lesion. But from the moment where, the period where we know that steatosis, microvesicular steatosis could evolve towards steatosis and cirrhosis, people began to be interested by the mechanism of this lesion. And recently with Julien Allard and collaborator, we made some investigation to know more about the mechanism that could be involved in this lesion. And to this end, we selected 12 different molecules. And in this investigation, performed in HepaRG cells, treated for 4 consecutive days with these 12 drugs. We wanted to have not too much cytotoxicity, and we choose concentration of each drugs that was able to decrease ATP or not, but if there was a decrease in ATP, not not above 30% of the basal level. That means that we always use concentration of drugs with a minimum of 70% of ATP levels within the HepaRG cells. So we tested the ability of these drugs to induce steatosis in HepaRG cells, and 9 of these drugs actually induce a significant increase in lipids levels assessed with the NIRAD fluorescence. And as you can see, there was a large variation between drugs regarding the intensity of this steatosis. And for example, amiodarone indomethacin and rifampicin induce a strong increase in the lipids within the cells, whereas not shown in these slides, but troglytazone, for troglytazone, this increase in lipids was moderate, whereas, I mean, even this was significant. So we wanted to know more about the mechanism, and beside mitochondrial beta oxidation, as previously explained, we wanted to evaluate de novo lipogenesis by using 2C14 acetic acids and by measuring its incorporation into newly synthesized labeled lipids that was extracted after a certain time of incubation. And we also measure VLDL secretion thanks to a proxy, in particular by measuring the apolipoprotein B, ApoB, in the cell supernatant, because there is one ApoB molecule per VLDL molecule. And so, as you can see, we use different positive markers to validate each of these measurements. And for example, for mitochondrial beta oxidation, we use atomoxir, which is an inhibitor of CPT1. For de novo lipogenesis, we use a mixture of stearat and oleate. Because when you add this fatty acid, the cells don't want no more to synthesize new fatty acids. So I mean the de novo lipogenesis, or DNL, is reduced. And finally, for VLDL secretion, we validate the assay by using lomitepide, which is an inhibitor of MTTP, which is a microsomal triglyceride transfer protein. And so, I mean, I will not show all the drugs, but just I choose 3 interesting drugs. First of all, amiodarone, of course, because again, this study confirmed that amiodarone is able to inhibit mitochondrial FAO. But as you can see, this inhibition was rather moderate for the 25 micromolar. Confirming that we did not select too high concentration able to strongly inhibit mitochondrial beta oxidation. So confirmation of the inhibition of fatty oxidation, but a moderate inhibition. And interestingly, we showed that amiodarone was able to increase de novo lipogenesis, in particular for the concentration of 15 and 20 micromolar. And this is interesting because previous article, in particular from our team, showed that amiodarone could increase the expression of SREBP1, which is a transcription factor involved in DNL. Regarding ApoB secretion, amiodarone was without effect on VLDL secretion. For indomethacin, it was interesting to observe that the 3 mechanisms could be involved in the occurrence of steatosis, in particular, slight to moderate inhibition of fatty acid oxidation, a large increase in DNL, and also a significant increase in ApoB secretion. And regarding troglytazone, we were very surprised to observe a strong inhibition of ApoB secretion, in particular with the dose of 45 and 47 micromolar. And interestingly, there was either an increase in fatty acid oxidation or a decrease in DNL, which are a mechanism that could not explain steatosis, but we made the hypothesis that the increase of mitochondrial FAO and decrease of DNL could be adaptive mechanism facing the large decrease in ApoB secretion. And in this article, we also assess the endoplasmic reticulum stress, because previous studies have shown a correlation between this ES stress and the impairment of VLDL secretion, and only show the results with indomethacin in these slides, showing that this drug was able to induce ER stress that was indicated by the high increase in the expression of several ER stress markers, such as BIP, SHOP, EREAN alpha, PERC, and ATF6. And regarding of IREA1, we showed that there was an increase in the protein level of IREAN alpha, total protein, and also the increase in phosphoirean alpha, in particular for the highest dose of indomethacin. And lastly, we made some experiments with TUDCA, which is a tauroursodeoxycholic acid, which is a chemical chaperone protecting against ER stress. And we showed that in the presence of 300 micromolar of indomethacin, the pre-incubation of the cells with TUDCA induced a significant reduction of neutral lipids, which was associated with an increased secretion of ApoB and also with an increased expression of the mRNA expression of ApoB. So altogether, I mean, this result and other showed that for some drugs, the reduction of VLDL secretion was probably, at least in part, induced by ER stress. In this study, we also showed that some of drugs, including amiodarone, increase sharply or more modestly the mRNA expression of fat CD36, which is an important transporter able to mediate the fatty acid uptake within mitochondria. And so we conclude in this study that microvascular steatosis, in the absence of severe mitochondrial function, could be a multi-mechanistic lesion with a role of a moderate or slight or moderate inhibition of mitochondrial beta oxidation, increased de novo lipogenesis, reduced VLDL secretion, and increased uptake of fatty acids. And so could be 1, 2, 3, or 4 of this mechanism for different drugs. As I told before, there are some drugs that can induce either microvascular steatosis or microvesicular steatosis. And this is the case, for example, with amiodarone, perexilin, etc. And it is unclear why some xenobiotics can induce in some patient, in rare patient, microvesicular steatosis, and in more patient macrovascular steatosis. But when we look at the literature, it is possible that underlying genetic disease might explain that in some case, people with underlying genetic deficiency in some enzyme could be more at risk for microvesicular steatosis. And indeed, some reports mention genetic deficiency in medium chain acyl-CoA dehydrogenase, MCAD, or deficiency in cytochrome C oxidase, and also in DNA polymerase gamma. And lastly, I would show the latest evidence for the importance of impaired mitochondrial fatty acid oxidation in drug-induced steatosis. In a recent paper that we made in collaboration with Methodologics, and in these papers, we select 32 drugs able to induce steatosis in patient, and 13 drugs that were never reported to induce steatosis in patient. And of note, these drugs could induce either microvesicular or macrovascular steatosis. We didn't discriminate between the two forms. And in order to evaluate the mitochondrial fatty acid oxidation, we study the potential of all these drugs to acutely inhibit beta oxidation in isolated mouse liver mitochondria, incubated with different substrates of the beta oxidation, namely either palmitoyl coenzyme A plus L-carnitine, or palmitoyl L-carnitine or octanoyl carnitine. And these three different substrates allow to know whether there is an inhibition of beta oxidation, and also where, meaning that depending on the results, you can say that the inhibition can be before CPT1 or after CPT1, and whether the inhibition, the oxidation of medium fatty acid oxidation can be inhibited. And in this study, but I invite you to look at the article. We showed that there was a significant correlation between drug-induced inhibition of mitochondrial beta oxidation, whatever the substrates, and the potential occurrence of steatosis in patients. And there was a rather good sensitivity and specificity in this test. So the take-home message as the following very quickly. So microvesicular steatosis is a mitochondrial disease, which is secondary to severe and prolonged inhibition of the beta oxidation pathway. This inhibition can be direct or indirect via an alteration of the respiratory chain activity, or even via an alteration of mitochondrial DNA homeostasis. And to explain microvesicular steatosis in a few patients, it is possible and demonstrated in some cases that the possible presence of underlying genetic disease affecting mitochondrial function. And regarding macrovascular steatosis, which is a benign lesion in the short term, we believe that it is a multi-mechanistic lesion with the possible involvement of different metabolic pathway, including beta oxidation, fatty acid uptake, de novo lipogenesis, and VLDL secretion. And finally, and I did not address this question in the talk, but it is possible that some polymorphism in gene, in particular in PNPL3, could predispose to macrovesicular steatosis. And there was a paper from Guzman in 2018 that could suggest this hypothesis. Finally, if you are interested by this topic, you can just read a recent paper. I've got the PDF today that I wrote with my collaborator, Annie Bon-Sanchez, and I can send you the PDF if you want. I thank you very much for your attention. I have been a little bit long, but there was an interruption in this talk, but sorry for that.
Yeah. Thank you very much, Bernard. And there is a good audience today, a very international audience. So I would like to encourage the audience to ask you a few questions before we finish. I hope that's okay.
Oh yeah, yes. I have a few minutes. I will be there.
Yeah,
Don't worry.
it
I
was
will. I
very
will not
clear.
escape. I will not escape.
I hope we won't have any more technical problems, though.
Yeah, I hope so. It's inconvenient. Yeah.
Yeah, please. Any questions from the audience?
Yes, Bernard, this is Santiago Lamas from Madrid, Spain. I really enjoyed your talk, and I have just two short questions. Can you hear me?
Yes.
Perfect. The first question is related to the fatty acid oxidation as a means to prevent drug toxicity. Do you know about the strategies that have been directed to increase fatty acid oxidation, let's say, towards the increase of function of CPT1A, or maybe through other strategies that can help to prevent or combat drug toxicity? And the second one is related to ER stress. There are some papers that propose that increased beta oxidation can lead to higher ER stress, which could be detrimental in the long term. What do you think about this concept?
For the first question, is very interesting, and in the recent review, we discussed about that, and in particular, there are some papers showing that L-carnitine could be interesting to prevent drug-induced steatosis and also drug-induced cytolysis. Because, I mean, it is very interesting also to mention that L-carnitine not only can enhance fatty acid oxidation, but there are also investigations showing that this beta oxidation cofactor can have anti-apoptotic effect and other interesting effect in case of liver injury. So, I mean, it is a possibility with some paper showing some effect and beneficial effect of L-carnitine in this for drug-induced steatosis. And also, we discuss in the review the possible but not shown yet possibility to test resmetirom, which is a tyromimetic, which is used currently for the treatment of steatohepatitis linked to obesity, and because these drugs can improve mitochondrial function and in particular beta oxidation, it will be very interesting to test, at least in the first time in experimental animals, to see whether resmetirom could be able to alleviate drug-induced steatosis. This is some example. And for your second question, because I don't want to be too long, regarding increased FAO and ER stress, yes, it is possible that ER stress in some case could be induced by increased oxidation of fatty acids, because I don't know if it is the exact mechanism, but some papers showed that some enzyme of the beta oxidation can produce hydrogen peroxide. This has been shown, for example, for VLCAD. And also there are some papers showing that some enzyme of the Krebs cycle in presence of fatty acids can produce large amount of reactive oxygen species. And so we can surmise that increasing sharply the production of reactive oxygen species by increasing fatty acid oxidation could secondarily induce ER stress. Thank
you very much.
This is one hypothesis. I'm not sure that this has been shown, but it is, for me, at the first sight, it could be interesting to test this hypothesis. Yeah.
We have another question from Ramiro Javier, please.
Yes.
Yes, thank you. Thank you very much.
Maybe quit the screen and see the audience instead. It might be better.
Switch the screen,
Stop
no?
sharing. If you stop sharing the screen, yeah.
Yes,
Perfect.
sorry. Sorry.
Okay, sorry, my video camera is also not working today. Thank you very much for the presentation, very nice. We in our group in Valencia University, we are working with other people in projects that want to predict the toxicity of drugs and chemicals. And they work a lot with the adverse outcome pathways concept, you know, this, and I think there is a discrepancy in these adverse outcome pathways regarding liver steatosis induced by drugs, because most of them are based on a molecular initiated event, which is the interaction of the drug with a receptor transcription factor, nuclear receptor or similar. And I am aware, and I confirmed today, because your nice presentation, that many effects of chemicals are not mediated by a preliminary binding to a nuclear transcription factor. You see these effects on the translation because there is an interaction with the ribosome or direct interaction with carnitine palmitoyl transferase 1. So this is the problem of this is that most of the regulatory people is just sticking to these adverse outcome pathways, and they will miss many mechanisms if you don't update the AOP wiki with this new or not new. I mean, this is the first mechanism were more direct effects of drugs, and now it seems that all the mechanisms are triggered by nuclear receptors, and this is a mistake. So I want just to commend that and to know your opinion.
Okay, so I mean, I think that the people are just mixing the drug-induced steatosis. When I mean drug, is pharmaceuticals, and steatosis induced by environmental toxicants. In the case of environmental toxicant, there is a large evidence that the activation of nuclear transcription factor or receptor can be involved. Regarding the drugs, there is not so much evidence that nuclear factors can be activated and responsible for steatosis, and because I mean, theoretically, you don't need to activate a nuclear receptor by drugs, because drugs are given at concentration and dose that very high. So I mean, they might be activated, but for the effect, you can explain the inhibition, some metabolic effect directly via an inhibition of enzyme. But also I recognize that for some drugs, for example, I cite the amiodarone example. Amiodarone could increase lipogenesis by activating SARBB1. So I agree that for drugs, maybe we should know more about the interaction of a nuclear receptor. But regarding the mitochondrial dysfunction, there is many investigation saying that it's a direct action on mitochondria and not via nuclear receptor. I don't know if I was clear in my answer, but
yes, thank you. It was clear, and it's very good point to take these concentrations, which are higher with drugs, of course. Thank you very much.
Could I ask a question, a small question? You mentioned at the end the possibility that some of the genetic risk factors for MASLD might be relevant to macrovesicular steatosis. Is there any evidence that people who have MASLD or perhaps MASH are more susceptible to toxicity, which presents as macrovesicular steatosis?
It is an excellent question, and we did write some review about this issue. I didn't want to address this issue today because it will be too long. But yes,
Yes.
there is some paper, I mean, some clinical investigation confirmed by experimental investigation showing that people with MASLD can be at risk for the toxicity of some drugs, not all drugs, but some drugs. This has been shown for some drugs. For example, for the antiretroviral drugs and others. And in the lab, we also work on the acetaminophen case because MASLD could, at least in some obese patients, increase the risk of acute hepatotoxicity induced by acetaminophen. Possibly via different mechanisms, including increased expression and activity of CYP2E1 in obese people, and also possibly by the presence of underlying mitochondrial dysfunction, even if this mitochondrial dysfunction is not necessarily very important in MASLD, depending on the stage. But you can find some review from our group and from other addressing this question.
Okay, I have one more question.
Yes, sure.
I work more on the immune-related liver toxicity, and we have reported that it is more common in older people, possibly because the concentrations in the liver might be slightly higher. Is that also the case with mitochondrial mediated toxicity? If it's a drug that causes mitochondrial toxicity, is it more likely to cause this in older people?
It is not shown. It is not shown. However, there might be a gender effect for mitochondrial toxicity, and this has been suggested in a clinical study from the Ayako Suzuki group. And this the article is cited in our review, in our 2025 review. I can send you the review. You will
Yes,
find.
please.
Okay.
That would be very kind. Does anyone else in the audience want to ask Bernard a final question before we finish?
And before the final curtain.
Exactly. Well, we've managed to get to the end without any more technical problems, which is good. So I think there will be some updates on this seminar series in due course by email. I don't know what is happening next. I'd like to also thank my co-chair, Marina, today, who had to go. And finally, thank everyone for being with us for this fascinating talk. Have a good evening, everybody.
Thank you very much for your attention.
Okay.
Bye.
Thank you.
Bye.
Bye.